Okay, good afternoon, everyone. My name is Zhang. I'm a PhD student of Shanghai Tech University. And first, I would like to thank the organizers of ISMB for giving us such a great opportunity to introduce our work. And today, I'm going to present our work named Knowledge Graph Reasoning for Explainable Prediction of Synthetic Lethality. First, let me introduce the background. Synthetic lethality is a type of genetic interaction. It means that two genes have a synthetic lethality relationship if the defect of one gene will not affect the cell viability, but the defects of both genes will cause the cell death. And this is a goldmine of anti-cancer drug targets because many cancers are associated with gene mutations, such as tumor suppressor gene. And for example, in this cancer cell, we know that gene A is mutated in this cancer cell, and we know that gene B is a SL partner of gene A. And we can kill this cancer cell by inhibiting gene B, but we can do not harm the normal cell. And additionally, if gene A is not druggable, then we can target the SL partner for the cancer treatment. So as we can see that SL is a very promising approach for cancer treatment. And the first thing we need to do is to screen SL gene pairs. There are several methods. For example, experimental screening such as RNA interference and CRISPR. This method usually have high cost and off-target effect. Statistical inference usually propose some hypothesis for SL interaction prediction, but this hypothesis usually require prior knowledge. So these issues speed up the development of supervised learning. This is a general training paradigm of supervised learning for SL prediction. We usually take a knowledge graph as the input, and this is actually a semantic network including many prior knowledge. And we use SL labels from a database as the ground truth. And using this data, we can train a network, for example, a graph neural network to predict new SL gene pairs. For example, this is a knowledge graph named SNSKG. This knowledge graph includes many gene functions and diseases and corresponding relations. And the KG4SL and POSL are two SL prediction methods. They randomly sample neighbors from this graph for genes and learn gene embeddings for SL prediction. But the random sampling neighbor, this method have some problems. First, the knowledge graph structures, which is a type of semantic information, cannot be fully utilized, and it also lacks interpretability. For example, in this knowledge graph, we can see that BRCA1 and ABL1 are both involved in DNA repair. And this is a very obvious connection in this graph. And we can use such connection to predict their SL interaction. However, if we randomly drop this node from the neighbor set of these two genes, such connection will not exist, and maybe we cannot predict their SL interaction correctly. And we also know that DNA repair is one of the key. It's one of the key functions of BRCA1, and maybe it may be very hard to explain the prediction without this node. So to address this problem, we can consider the path-based knowledge reasoning. Knowledge reasoning is to predict new links by existing knowledge. And here we can regard SL as a type of knowledge, and we can use knowing SLs and a knowledge graph to predict new SL gene pairs. And path-based knowledge reasoning is usually performed by relational paths. Relational path is a sequence of relations acting like rules. And using such relational paths, the reasoning process will be interpretable, and we can also perform inductive prediction on new entities. This is a heterogeneous graph with directed edges, and we add knowing SL in this graph, and we can see that there are many passes between TP53 and the CDQ1. For example, this pass tells us that one of the SL partner of TP53 and the CDQ1 both participate in DNA damage response. And we can further convert this path into relational paths and use such relational paths to infer their SL interaction. But there may be multiple paths connecting the two genes. So here we introduce a more complex structure named the relational directed graph, also known as relational digraph. This concept is proposed by a previous study. For example, this is a relational digraph for TP53 and the CDQ1. This graph has only one source node and target node, and it preserves all relational paths with all neighbors, and all the paths have the same length. And we can see that such graph structure is more informative than single relational paths, so we can use such graph structure to predict SL interaction. Next, we formulate the SL prediction problem as a gene ranking problem. So given a primary gene, GQ, we use the relational digraphs to predict the possibility scores for all candidates, and we rank all unknown candidates, and we take top K candidate partners as the output. This is an overview of our framework. First, we construct a heterogeneous graph by knowing SL graph and a knowledge graph. And then, based on this heterogeneous graph, we can extract all relational digraphs for all the gene pairs. And here, for the relational digraphs with the same source node, we can take the union of these digraphs and learn the semantic embeddings. And finally, for the genes at the last layer, we can calculate their scores and get ranking as the output. After we finish the prediction, we can extract such subgraph from original graphs according to the attention scores. And such graph can reveal the important prediction process. Next, I will introduce the details of our method. The overall reasoning process is performed within relational digraphs. For a primary gene GQ, we search the relational digraphs for all its candidate partners with key steps. For example, in this graph, we take TP53 as an initial source node, and we find all of its neighbors as the target nodes. And then we perform the message propagation from this source node to all its target nodes. And actually, here we find all the relational digraphs between this source node and the target nodes. And then we take these target nodes as the new source nodes. And based on the previous relational digraphs, we can find the new, more large relational digraphs between this source node and these new target nodes. And we repeat these steps for k times. And finally, at the last layer, we can get the target nodes at this layer. And the genes within these target nodes are the candidate partners of this TP53 gene. The semantic information is propagated along directed edges. And here we introduce three types of semantics. The first is textual semantics. Here we take the entity description as the input and use a BERT pre-trained on UMLS to extract the textual embedding for each entity. And the structural semantics is captured within the relational digraphs from the source primary gene to a current node. And here, for one of the triple EI, RIO, EO, we calculate the semantic embedding using 4 terms. The first term is the semantic information propagated from GQ to EI with k-1 steps. And the second term is the relation embedding of RIO at case layer. And TI and TO are the textual embedding of EI and EO respectively. And as we can see that there may be multiple source nodes connecting to EO, so we adopt attentive aggregation to calculate the semantic embedding of GQ-EO at case layer. Here we can see that actually TI and TO, which are 2 textual embeddings, are fixed during the learning. So the structure semantics is mainly to learn the relation embedding. And finally, we further enhance the sequential semantics of relational paths using a JRU module. And finally, we can get the semantic embedding of GQ EO at a case layer. And at the last layer, for a candidate gene GP, we pass the semantic embedding through a feedforward layer to get the scores. This score means that a possibility of GP being an SL partner of GQ. And to optimize our model parameters, we use a multi-class loss function, which actually is a cross-entropy loss. And in this function, for a training gene pair, we normalize the score across all the gene pairs with the same source gene. So this is all of our method. And next, I will introduce the experiments. In our dataset, the SL graph and SL labels are from CLSDB. This database is a very comprehensive database about synthetic lethality. It includes many SL gene pairs from wet lab experiments or computational prediction. And it also includes a knowledge graph, CLSKG. So the knowledge graph we used is from both CLSKG and the ontoprotein. And the final heterogeneous graph has 33 relations. Here we design 2 types of experimental scenarios. The first is transductive reasoning. In this setting, we predict the missed SL partners for a gene. But in inductive reasoning, we predict SL partners for genes unseen during training. For example, in this illustration, we can see that in transductive inference, the graph is the same with the training graph. But in inductive inference, the entities in this graph are different from the training graph, but the relations are same with the training graph. We adopt 3 types of ranking matrix for the evaluation. And the higher value means better performance. And we also we use 8 types of SL prediction method as a baseline. And these baselines use different type of input data. And we can see that our model performs the best. One of the reasons is that our model combines both knowing SLs and knowledge graph. This is an inductive reasoning result, and we can see that most of the baseline methods have poor performance. This is because that this method is mainly to learn gene embeddings from a graph for prediction, and a new gene without training cannot be predicted correctly. But our method is mainly to learn the relation embedding, so it can still perform well on new genes. And we also conduct several ablation studies. The first is to test the effect of randomly sampling neighbors. So to do this, we sample several neighbors randomly for each node at each layer. And from the result, we can see that as the number of neighbors increased, the performance also increased, but all of these models cannot exceed our model. So this means that randomly sampled neighbors will lose graph structures that are beneficial for the SL prediction. And we also conducted 3 model variants without textual information or attention mechanism or GRU module. And we can see that compared with original model, the result of these 3 variants both decrease. So it means that first, textual information is beneficial for SL prediction. And the SL mechanism can help capture critical structures for both prediction and interpretation. And finally, we can find that removing GRU module significantly decreases the model performance. So this means that the sequential dependency of original paths is very essential for SL prediction. Finally, we visualize explanation subgraph between BRCA1 and USP1. This subgraph is composed of directed edges with significant attention scores. And in this subgraph, all the passes follow a same rule. This rule tells us that the SL partner of this BRCA1 and the USP1 are both involved in some gene ontology node. And such rule can reveal important prediction process. And we know that BRCA1 is a very famous cancer driver gene. And recently, USP1 has been found to be an SL partner of BRCA1. And actually, I remember that in Silicon Medicine has reported that USP1 is a very promising SL target. So this is because the function of unicombination for DNA repairment. So these functions are also consistent with the gene ontology node in our subgraph. So our subgraph can provide evidence for SL mechanisms. In summary, we highlight the importance of using relational diagrams, and we propose a path-based graph neural network method for SL prediction. And our explanation subgraph provide both interpretability for SL prediction process and the potential SL mechanisms. And in the future, we first plan to divide SL interactions according to the cell lines so as to generate the context-specific explanation passes or explanation subgraphs. And as we can see that the textual information is actually beneficial for SL prediction. So in the future, we also plan to combine the knowledge graph structure and the language model, such as GPT, to generate the natural language explanations for SL mechanism. And finally, I want to thank Professor Yang from School of Life Science, Shanghai Tech University. She is a biologist, and she gave us many insightful suggestions for making explanations. And I also want to thank our students from Cell Lab. Their previous method gave me many inspirations of designing the model. Okay, that's all. Thank you.
Very nice talk, Zhang. Thank you very much. Is there any question from the audience?
Thank you. I'd just like to ask a little bit more about your benchmark data set. So SynLethDB has a score for each pair that's used. Do you know what threshold you used or what distribution of scores you had for your benchmark data set?
You mean the quality of the data set?
Well, you know, SynLethDB has a score for every synthetic lethal pair, which relates to the different evidence that's used to, for example, you mentioned that a lot of your SynLethDB has computational predictions as the evidence for the pair, for the synthetic lethal pair, and because that's only based on a computational prediction, then you could imagine that if somebody had used a gene ontology to make that prediction, that would obviously bias very much in favor of a gene ontology-based method such as your own. So just thinking about the score, like, would you know if you use a threshold or what the distribution of?
Yeah, actually, in this work, we do not consider to filter the scores lower than a threshold. We just regard all of the gene pairs in the database as the positive labels. Actually, the computation of the scores of ScienceDB is super simple, and maybe it may be not very aligned with the actual experiment, I think we cannot find a accurate or accurate approach to evaluate whether these scores are really aligned to the experiment or not. So we just regard all the gene pairs as the ground truth.
It would be interesting to maybe increase the threshold value.
Yeah.
For example, a value of 0.7 is perhaps one to consider where you do have experimental evidence above that value.
Yeah, that is a necessary future work. I think why we use all the data SL pairs is because that we need to train a model that such expressive model usually require a lot of data, and yet the size of true SL gene pairs are not very large, so we just want to use as more SL data as possible. Yeah.
Thank you.
Oh.
Thank you. Are there
Thank you. That was a really interesting talk. I was wondering whether you experimented with different values for K and whether you saw that your graphs would explode and sometimes it would work less or
You
less well or better.
you mean top K?
K for the number of steps when you create your subgraphs.
Yeah. Here we only show the result of 3 steps, and actually we perform the result using the K from 1 to 4. And if you read the paper, you can find that the result of 3 hopes is comparatively with the result of 4 hopes. So I think maybe 3 hopes already cover most of the gene pairs. Yeah.
Brilliant. Thank you.
Thanks, this is a very nice talk. I was curious if you could explain how you trained your BERT UMLS model, because that already has explicit semantic structure.
Yeah, we use wait a minute. We use a BERT to extract the textual embedding, but actually we do not train the BERT together with our model. We just use the pre-trained model to take the embeddings and fix the embeddings, fix the textual
So
embedding.
you used an existing pre-trained BERT.
Yeah, this is a pre-trained BERT.
Okay, but you don't know how that was pre-trained.
Yeah, right now we don't further train the BERT together with our model, but I think this is the future work we can do.
Thank you.
I was wondering, like, thanks for your great talk, by the way. I was wondering, since you were the previous slide you were showing, where you were kind of using the GRU to then embed the whole trajectory. If you would like now compare all the positive pairs that you find and you kind of cluster them, did you try something like that to kind of see, hey, well, this is like, it was not actually this, you know, you had one where you had like BRCA1 and then PARP, and then this is obviously one mechanism, but there must be different mechanisms of a landscape kind of this synthetically lethal possible kind of interactions, right? You could now kind of cluster those. Maybe that would be interesting. Did you try something like that?
Yeah. You mean I can first perform the data analysis such as clustering to see their similarity.
Yeah, to see whether there's different groups kind of synthetically lethal interactions, you know, because now you have basically everything to do that, right? You could now make a picture of, okay, this is a whole group of things that involve DNA repair, and you know, and this is maybe another mechanism.
I think this can be do after the prediction or the explanation. Yeah, because we actually want to find the similarity between the new predicted gene pairs and the existing SL gene pairs. So after we find the such graph font pattern automatically, and next we can do the clustering to further find their common functions.